Respected organizers and eminent scholars, my name is Rahul, and today I am going to briefly present a very simple yet valuable work from our lab titled Cultivation of Pleurotus florida by Standard Protocol, in a study of the temporal changes in total protein contents of this oyster mushroom relative to growth of its fruiting body. Proteins are macromolecules formed by amino acids. Proteins are the ultimate players in the processes that allow an organism to function and reproduce. An average adult requires 46 to 56 grams of proteins per day. It is unfortunate and a sad reality that 25% of all hungry people worldwide live in India. In India, 44% of children under the age of 5 are underweight. Out of these underweight children, many suffers from protein energy malnutrition diseases like marasmus and kwashiorkor. The conventional sources of dietary proteins are fish, meat, eggs, milk, pulses, soya bean, peas, cereals, etc. Mushrooms are one of the richest non-conventional sources of proteins. These are easy to grow and can contribute positively in facing the challenge of worldwide food shortage. We know that DNA codes for mRNA and mRNA codes for proteins, and the amount of protein in living organisms is not constant throughout life. This is due to time-based differential expression of genes, which is known as temporal gene expression. Keeping this information in mind, we cultivated oyster mushroom Pleurotus florida using a standard protocol and determined the temporal changes in its overall protein contents related to growth of the fruiting body. In this regard, this is our work plan. Pertaining to site selection, we chose ICAR Holy Cross Krishi Vigyan Kendra, situated at the foot of Canary Hill, Hazaribagh, Jharkhand, for mushroom culture. We had selected Pleurotus florida for our study because it has smaller generation time, can be grown on a variety of substrates, does not require much space, can be grown whole year except hot summers, and its cultivation does not require much financial input. For cultivation, we started with obtaining pure culture, spawn preparation, and spawning followed by cropping and harvesting. In this slide, I have shown a worker holding fully infested or mature spawn in his right hand compared to freshly inoculated spawn in his left hand. This is the image of our culture setup displaying fruiting bodies of the mushroom in different growth stages. Note that we have harvested mushrooms from stage 4, 5, 6, 7, and 8 for further studies to assess temporal changes in total proteins in our lab. In further studies, we will be referring these stages as stage 1, 2, 3, 4, and 5 respectively. As shown in this slide, we measured the size of the pileus and moisture content of equal weight of pileus from different growth stages of the mushroom, followed by estimation of its protein content. This is how we measured the moisture content. Here we find that both moisture content and size of the pileus increase with successive growth. For estimation of total protein content, Barrett method was used, which is a very simple and easy to use method based on colorimetric analysis of the intensity of color change in the sample. In this method, a calibration curve is plotted based on known concentrations of bovine serum albumin or any other protein in linearly ascending order. The concentrations of total proteins at individual stages were elucidated by extrapolating their individual OD values on the calibration curve. Total protein contents at individual stage, when plotted together, show a decreasing trend per unit volume with respect to the growth stages. Further, transverse sections of the pileus at stage 1 and stage 4 revealed development of large water-filled intercellular spaces with less cell density or number per unit volume in stage 4 with respect to stage 1. Anatomically, most fungi acquire large intercellular spaces at maturity, which accounts for its sponginess. In this slide, the spaces are indicated with red arrows. Based on these studies, we conclude that there is a gradual decrease in overall concentration or amount of proteins per unit volume with the growth of the fruiting bodies in Pleurotus florida with increase in size and moisture content. We assume this to be true for most of the mushrooms. We know that proteins form the major proportion of cytoplasm, hence more the number and density of cells in the lower stages, more is the amount of cytoplasm, and hence more is the content of proteins in the mushroom at lower stages. These are the references. Thank you.